It's an immense honor and pleasure to welcome now to Living Histories Professor Rosemary Braun from Northwestern University. Please take it away, Rosemary.
Hi. So thank you so much for the opportunity to present. I've followed Living Histories for years at this point, and it's an immense honor to be able to give one myself. So thank you for this. So I'm faculty at Northwestern University, and I also have affiliations with the Santa Fe Institute and now the National Institute for Theory and Mathematics in Biology. I call myself a theoretical or a computational biologist, but I didn't start out that way. I started out as a physicist, and so I'll tell you a little bit about that trajectory. But in order to set the stage, I want to tell you about the things I'm interested in now, which is how living systems self-organize. And I've been fortunate over the course of my career to study self-organizing dynamics from a mathematical and statistical perspective at all scales, from the atomic level, where proteins self-assemble into macromolecular complexes, all the way up to the organism and the ecosystem level. So as I mentioned, I started out as a physicist. I did my undergraduate work at Stony Brook. And when I got there as a young physics major, I took a class in Stat Mech from Jerry Braun, who's at the center of this picture here. Um, and it was the most beautiful thing I had ever seen. It, you know, it gave us a way to connect what happens microscopically to what we observe macroscopically, and I fell in love with it. And so when I was looking for a honors thesis for my undergrad, I immediately gravitated to Jerry since he had taught this class that I had so enjoyed. But Jerry was not working on condensed matter or anything related to statistical physics. He was working on problems in astrophysics. And so I became part of the nuclear theory group working on a project having to do with the evolution of binary pulsar star systems. It was a pretty typical undergraduate research project, which means it didn't go very far. It was mostly a learning experience for me, but it was a transformative one because this group of individuals, the faculty and the grad students who were part of the nuclear theory group at Stony Brook, really kind of became a scientific family. I was a young female undergraduate in physics in the mid 90s. I was definitely in the minority. And at no time in this was I ever made to feel inferior or not fully supported. It was a really remarkable environment, and I was very, very lucky. And the people that I met, including Andy and Adam and Maylee in that group, remain close friends and trusted advisors to this day. So I feel very fortunate to have had that experience. When I went to graduate school, I thought I would continue to do astrophysics, but then I took another class, again, a statistical mechanics class, this time a non-equilibrium stat mech class, and once again fell in love with the topic. And so I joined the lab of Klaus Schulten. So this is Klaus here. He was my professor for the non-equilibrium stat mech class. And of course, when you study non-equilibrium statistical physics, all of the examples are from living systems, because for something that is alive, to be in equilibrium is to be dead. And so that drew me into questions in biology that I thought I wasn't interested in, right? To me, biology was something where you memorized parts. But now, seen through the lens of statistical physics, it became fascinating to me. And so my graduate work had to do with simulating and analyzing the self-organizing dynamics of this little system that you see here, which is an SDS micelle solubilizing glycophorin A transmembrane helices. And so starting from a completely random configuration of these atoms, you can just simulate Newton's equations of motion and watch the self-organization take place. And at the end of this simulation, what you get is a very highly ordered system where the SDS molecules have all arranged themselves so that their hydrophobic tails are pointed toward the transmembrane helices, and the polar head groups are on the outside, and it's excluded water from the interior. And we were able to see this happen at atomic level detail, which was completely wild to me, and very inspiring. And so I thought, as I was completing my PhD research, that I would continue to do this type of molecular biophysics work, but I wanted to do it in a setting or in a system which could impact people's lives and people's health. Glycophorin A is a very well studied uh, system, which is why we were interested in simulating it, because there was plenty of experimental data to compare to. But it's not going to cure any diseases. And so I looked around for a postdoc that might help me move in a more applied direction. And I found this absolutely incredible postdoctoral fellowship that's run by the National Cancer Institute. It's called the Cancer Prevention Fellowship Program, and they recruit PhDs from all sorts of different disciplines, from physics, like me, to molecular biology, to behavioral science, to things like linguistics and medical ethics, just really all possible disciplines across the board. And the goal is to bring people in from all of these different disciplines and set them loose on the problem of preventing cancer. And because they know that they are recruiting a bunch of people with no knowledge about cancer research at all, the very first year of your postdoc, they send you off to do a master's in public health. And while you're doing your master's in public health, you're being paid as a postdoc. And so it's kind of nice in that respect. And then when you're done with the MPH, you get to join any lab that you want within the National Cancer Institute, and you're funded through the fellowship. And you get actually a little pay bump because now you have an additional degree. So it's kind of a nice program in many respects. But for me, it was transformative because when I was doing that master's, I found myself as a physicist in a school of public health, wondering what I am going to do for my master's thesis. Um, and at the time, this was when high throughput technologies were revolutionizing molecular biology. So rather than studying one gene at a time, we could now measure the expression of 20,000 genes simultaneously. But of course, what we want to know is not what's going on down here at this microscopic scale of 20,000 genes. What we're really interested in are things like, is this likely to become cancer? Or will it respond to a drug, right? Macroscopic observables. And I, being a very arrogant young physicist, decided that this was basically a stat-mac problem, right? We want to link microscopic interactions to macroscopic observables. I found out that it wasn't quite so simple, and I spent a long time at the National Cancer Institute learning genetics and statistics of a sort that really we don't get exposed to as physicists, and really delve deeply into that problem, and that changed my career trajectory completely. And this is this question of how do you link the microscopic to the macroscopic using high dimensional molecular biology data is a problem that I remain interested in till today. During my postdoc, I finally had a chance to go to the Santa Fe Institute as a student in the complex systems summer school. And this had been a dream that I had since first hearing about the complex systems summer school when I was an undergraduate. And I never got to actually attend until I was a postdoc. But again, it was a transformative experience. And while I was there, I started working with Dan Rockmore and Scott Pauls and Greg LeBon. on a problem related to spectral graph theory, which is a very elegant framework to connect the structure of a graph to dynamics of processes on that graph. And I realized that this could be a way to connect our microscopic data to macroscopic observables via what we know about the structure of molecular interaction networks. And so when I first came to Northwestern as a young faculty member, my first NIH grant was to use spectral graph theory as a way to summarize gene expression on a network and be able to say something about the behavior of whole networks rather than individual genes. And we were able to apply these techniques to discover a number of things, including things like gene signatures that can predict adverse reactions to radiation therapy in cancer patients. The other thing that happened by chance while I was at Northwestern was that I met a bunch of researchers who are interested in circadian and sleep medicine. A wonderful group of people, including Phyllis Z, Ravi Alada, Kathy Reed, Saber Abbott, Fred Turek, the names go on and on and on. And that opened up a whole new research avenue for me related to biological timekeeping. I learned from them that every cell in your body is running a molecular clock. And these clocks obviously need to be internally synchronized as well as synchronized to the external environment. And so we started studying sleep and circadian rhythms, both from the omics perspective, analyzing high dimensional data, and also just modeling them as systems of coupled oscillators. And these types of models enabled us to make predictions about the optimal meal timing for recovery from jet lag. And so on the basis of some of our models, we were able to say, well, the best thing you can do when traveling to a new time zone is to eat not when you're hungry, but on the time of that new time zone, and ideally to eat a big breakfast. So our model suggests that when you arrive in Paris, your very first breakfast should be as many chocolate croissants as you can handle. Um, and so I would say that it's been a fairly meandering path. But if I had lessons for young people from all of this, I would say the first lesson is to always be open to new things. Variety literally is life. Every path that my career has taken has occurred through chance meetings with individuals who said, hey, here's a funny, interesting problem, and got me interested in it. And we just sort of chased it. And that brings me to my second lesson, which is that great collaborators, the best collaborators that you will have will be people who are your science buddies, right? People who are your friends, who you just want to play science with. You know, we talk about playing music and playing sports, and people who do those things professionally, they do them seriously, but we still call it play. And the best science, I think, is the science which is playful. And so cultivating that is it's a wonderful thing. It's been very fulfilling for me and also very productive. And the third thing that I would say is that all of those chance paths were only possible because of a foundation that I had built with the help of others previously. Chance always favors the prepared mind, and you never know what knowledge you might gain now that will help you in the future. And to students who feel like, oh, I don't know why I have to study this, it doesn't pertain to the things I'm interested in. I felt that way too. And later I discovered that sometimes those things came in handy in ways I didn't expect. And so those are the lessons that I've learned over my career. And so that's my history. I need to thank the wonderful, wonderful group that I have the privilege of working with, some but not all of whom you see here. And they really they're why I enjoy what I do so much. So thank you.